So next up, we have Palani Akana going to be talking about winning the lottery of forest soil. Nitrogen availability is concentrated in hotspots, and some seedlings strike it rich. Take it away, Palani.
All right, thank you very much, Katie. Okay, so yeah, this project sort of got started. So this was also a project funded by BlackRock Forest and completed this last summer during the pandemic up in the field at BlackRock. And sort of the motivation for this study was reading a lot of papers about soil nitrogen sort of being this really important resource for trees, but really also a variable resource. And some studies finding that soil nitrogen within a forest could be as variable as soil nitrogen between forests or between different biomes. But this sort of like small spatial scale variability in soil nitrogen is something that isn't very widely studied. And so my goal for this past summer of research at BlackRock was to investigate on quantifying the small scale spatial variation in plant available soil nitrogen, and then extrapolating that to what that means for seedlings in terms of seedling nitrogen access and seedling growth. So here's a map of one of the few studies that have looked at this, the variability of soil nitrogen. It was done in a Canadian beech maple forest. And yeah, the plot here is just showing a map of soil nitrate, so one of the predominant forms of plant available soil nitrogen, on a 50 by 50 meter forest plot. And so it's widely variable and sort of concentrated in hotspots. You might imagine that as you have seedlings and trees growing up, if they're going to be found in a hotspot where maybe you have 12 milligrams of nitrate per liter of soil, that growth of that seedling is going to be much, much higher than a seedling that's happens by chance to be growing in this patch where there's only 1 milligram of nitrate. And so I really wanted to quantify this more rigorously, and BlackRock Forest was a really good site to do this. And so in this project, I'm asking two questions. The first one is, what is the spatial distribution of soil nitrogen, both in terms of availability and supply, and I'll explain what both of those terms mean in a minute, in a temperate forest, and then extrapolating that to tree access to soil nitrogen. So how does the spatial distribution affect tree access? So sort of the goal is, again, to analyze the spatial variation, to characterize soil nitrogen hotspots, and to extrapolate this to tree nitrogen access. So I conducted this research in two plots in BlackRock Forest, one sort of a typical BlackRock Forest composition where it's mostly red oaks, a few white oaks, a few red maples, and this other plot that was about 50% of the biomass in black locust, which is a nitrogen fixing tree, kind of early successional, one of the few species in temperate systems that can, through bacteria, as in the last talk, bacteria sort of taking nitrogen gas from the atmosphere and then incorporating it into the plant tissue. And so often these plants will grow in early successional environments, and sort of using these as two different ways to set. So what's occurring in a typical forest with soil nitrogen, and then what's occurring in an early successional nitrogen fixing dominated forest. And in each of these plots, I collected 155 cores from approximately 20 by 20 meter area. And then the distances between cores were between 10 centimeters and 20 meters. So I'm really honing in on this sort of sub 20 meters fine spatial scale resolution. And then here's a picture in the black locust plot to give you an idea of what that looks like. So, the trees are reasonably big by this point. And each flag is marking a point of the soil cores. So you can see there's some spaces where there are not many flags, and there's some spaces where these flags are clustered together. And that's just sort of this staggered spatial design of the sample in order to get both wide ranging, but also very close distance measurements of these soil nitrogen availability. To give you a picture of what the field work actually looked like, picture on the right is me with a soil core. So I sort of pound this into the ground to about 10 centimeters. And then at each of these 150 or so points per site, I'm taking two soil cores. One is being analyzed right away for plant available soil nitrogen. And so soil nitrogen in the ground exists in several forms, but inputs come in organic matter. So as leaves are falling from the trees, they enter into the soil. Most of organic nitrogen is not accessible to plants, but bacteria and fungi, microbes will decompose this primarily into ammonium, or NH4+. And that is accessible to plants. And then there are another group of microbes that will turn that into nitrate, or NO3, which is also available to plants. And so this, a lot of people, if you're looking at nitrogen in soils, will look at nitrogen availability as what's available to plants at one point in time. And so that's just your ammonium plus your nitrate. And then a second measure is what's known as nitrogen mineralization, which is more akin to nitrogen supply. And so that's the nitrogen that is made available through time to plants. And for that, we need a second core. And that second core is taken at the same time as the first core, wrapped in saran wrap, put back in the ground, left for about 2 months. And then the ammonium and nitrate pools by then will have increased. The main way that these ammonium and nitrate leave the soil is through plant uptake. And so by taking that core and severing the root connections, putting it back in the soil and allowing ammonium and nitrate to accumulate through time. And then I can also get a measure of final ammonium and nitrate, and subtract the initial from the final will give me this measure of nitrogen supply. So both availability and supply are metrics that are commonly looked at when thinking about this work. But I think really supply more accurately reflects the sort of plant available soil nitrogen, because it's more this time integrated measure of nitrogen that's being made available to plants through time. So here we just have a histogram of nitrogen availability in the two plots. You can see the non-fixer, the oak-dominated plot, tended to have higher nitrogen availability. So this is at one single point in time. And this I sort of explained by the fact that the Robinia, or is likely explained by the fact that the nitrogen fixer-dominated plot is growing a lot faster. And so even though these trees can provide a lot of their nitrogen from the atmosphere, they also need a lot of nitrogen and they're growing very fast. And so they're drawing down nitrogen within the soil. And in contrast, we see the opposite pattern between sites if we look at nitrogen supply, where the nitrogen fixer plot has higher rates of supply relative to the oak plot. And this is because you have these nitrogen fixers that are providing nitrogen from the atmosphere, this really nitrogen-rich tissue that then becomes decomposed and made available kind of within the ecosystem. And so you can also see there is this wide range in both of the plots of both availability and supply. And so this starts to get at some ideas of hotspots and like how concentrated is the soil nitrogen in different hotspots. The next plot that I'll show is just some maps of the two sites. So again, here we have nitrogen availability, the oak site and the Robinia site. These are just meters by meters on the X and Y axes. The red color is showing more nitrogen available. So that's what the availability looks like. And then the supply, you can see on a different scale, but sort of the red colors again showing where this nitrogen is concentrated. And so I'm going to sort of move into this, the rest of the talk is talking about spatial distribution of soil nitrogen. And I want to focus in on two ideas or two metrics. The first being this inequality. So inequality is really asking how concentrated is the soil nitrogen in hotspots, and spatial autocorrelation being, well, what is that spatial scale of correlation? Is that a few centimeters, a few meters? If you go and you take 1 point, measurement of soil nitrogen, does that tell me anything about a soil nitrogen measurement 5 centimeters away, 2 meters away, 10 meters away? So what is that spatial scale? And so just from these maps, we can start to get a little bit of idea of this kind of spatial autocorrelation picture, where we can see these red high nitrogen availability points tend to have other high nitrogen availability points next to them. So saying these hotspots might be, on the scale of 50 centimeters to 1 meter, versus nitrogen supply, it appears to be smaller hotspots. So maybe 10 to 50 centimeters, I'm not really going beyond that. And this is just saying visually from these plots, but I can also analyze this more rigorously. And so I'm going to first delve into inequality and spatial autocorrelation, and then come back at the end to say, what does this mean for tree seedlings? So here we're looking at the inequality of our two sites shown in different colors, and as well as the two metrics, so supply and availability, shown by the dashed and the solid lines. And x-axis showing the percentage of soil patches, and then the y-axis showing the cumulative percentage of soil nitrogen. And so you might have also seen these, they're called Lorenz curves. Sometimes people look at them for like inequality of wealth within a society. But basically, the further the line is away from the one-to-one line, the more unequal the soil nitrogen is. And how you can read these plots is if you come up and say, well, I'm going to look at 50% of soil patches, go up from the x-axis, and then where that intersects, I say about 10% of soil nitrogen is contained within 50% of patches if I'm looking at the supply of the oaks. And the other 90% is contained within the other 50%. So that's pretty unequal. If we're thinking about hotspots, then maybe we would take 10% of soil patches. And for all of those, so that's this black line drawn here, for all of those, about 30 to 45% of soil nitrogen availability and supply is contained just within 10% of soil patches. So then going to the second metric, this is a spatial autocorrelation. How this is often analyzed is through what's known as a Semivaryogram, which is shown here. So on your x-axis, it's distance between pairs of soil cores, pairs of points, and the y-axis is showing the variance. These plots are a little confusing, so I'll sort of walk through how they're generated. But essentially, this 1 point would be for all pairs of soil cores between 0 and 24 centimeters. So maybe one core I have a measure of 5 and another core a measure of 20. And then I'll look at two other pairs that are maybe also 10 centimeters away from each other, and I get 13 and 21. So for each of these, for all of these pairs, I'll then get the difference. So the difference between 5 and 20 is 15. Do that for all, say, 30 to 40 points. And then I can get a variance of that. And that variance is what gets plotted up on that graph. And then for the next point, we would do the same exact thing. So each of these points are from at least 30 pairs of soil cores. And then we can do the same thing for both nitrogen availability at both sites and nitrogen supply at both sites. And sort of what I would say to notice here is that we do see this spatial structure in availability, but not in supply. And so especially for less than 2 meters, that's sort of where your hotspots are, where you have more information about a neighboring soil core based on a soil core that's already there. But when we're thinking about supply, even if you're moving 10 centimeters away within the forest floor, you can't really say anything about what the supply is based on what it is 10 centimeters away. There's basically no spatial structure in that, which is, I think, a very interesting finding. So the last point that I want to make is extrapolating this out to tree nitrogen access, sort of combining these metrics of inequality and spatial autocorrelation. And so if we just, and these are done using through simulations, looking at sort of one inequality line that could represent either availability or supply, and then going up in spatial scale from 5 centimeters to 25 centimeters to 200 centimeters, and thinking about this as a rooting diameter of a plant. And so nitrogen availability for a plant that has a 2-meter rooting diameter is still having a somewhat unequal distribution of soil nitrogen. And that largely has to do with the spatial autocorrelation. If we were to take that same initial black line, but now get rid of the spatial autocorrelation, as we see for nitrogen supply, I'll add those lines in here in the dashed. Essentially, by the time you're at 2 meters and a rooting diameter, you have pretty much anywhere you're growing in the forest as a tree, you would have equal access to soil nitrogen. And to sort of put this in perspective, maybe a 25 centimeter rooting diameter would be a tree that's 6 to 12 inches tall, so a very small seedling, versus something that's 200 centimeters or 2-meter rooting diameter might be a tree that's 5 to 10 feet tall. So here we're still talking about seedlings and saplings, saying that the soil nitrogen inequality, the spatial distribution is really important at these small spatial scales. So just to summarize, you have plant available soil nitrogen being very concentrated in hotspots, with 30 to 45% of this nitrogen in about 10% of soil area, and availability but not supply being spatially correlated. And then the trees, the seedlings, have much more variable access to soil nitrogen, with a patchiness in soil nitrogen likely having a large impact on the growth and survival of young trees. So seedlings that are, maybe in the range of 1 to 10 feet tall at the most. So thank you all very much for listening. Thank you to my advisor, Professor Duncan Menge, as well as all these other people and funding sources listed on this slide. I especially want to thank the following people listed here from the Black Rock Forest staff for lots of support with the summer research, and also the David Redden Conservation Science Fund grant. So thank you all very much. And I think we're probably out of time for questions, but I'll be around in the room later on to answer anything. So